Okay, hello everybody, and did not see you this last Monday. I just want to remind you that you have your midterm test is on this coming Monday. It does not include the material which we're going to discuss today. This will be on the next test. Your test on Monday only includes metabolism, and that is both sections, including the photosynthetic processes that you did on your own on the HHMI exercises. Please remember that that is examinable, and I will be sending you details of a short written portion on your midterm, which relates to that. Okay, so in our section on metabolism, we discussed at length those processes of metabolism. First of all, the catabolic reactions, which derive energy into biologically useful energy, and largely in the form of ATP. That energy is used in the anabolic reactions, which are the building reactions, building biomolecules. Why build biomolecules? First of all, you may want to build biomolecules to replace molecules which are worn out, which need to be repaired or whatever. But second of all, growth is a feature of all living things. All cells grow. They grow to a certain size and then divide under carefully programmed cues in DNA. Growth in bacteria in particular results in division by binary fission. One cell grows to a certain size and then divides. So this process of growth is something which we need to discuss in detail, because we're interested not only from a biological point of view, in the processes of growth, what cells need, how they grow, etc. This is also extremely important in clinical settings. We need to understand how bacteria grow in order to deal with them under different clinical settings. So let's have a look first at the physical requirements for growth, because you'll realize that all of the metabolic processes we dealt with were governed by chemical reactions. Those chemical reactions catalyzed by enzymes. And this in particular is of importance because the enzymes have strict requirements depending on the enzyme, and especially depending on the species of bacterium where they live. The enzymes which govern the rates of metabolic processes actually may differ depending on the enzymes and depending on the species of bacterium. All enzymes, as we saw before, have an optimum, a range of conditions where they operate best. And that is called the optimal, and enzymes have optimal temperature, optimal pH, and in addition, these all add together in the processes of metabolism to produce a requirement for different bacteria. Different bacteria have different optimal temperatures they grow at because of the enzymes. Different pHs, optimal pH, for example, that they grow at because of their enzymes. But the whole organism itself may have conditions upon which it depends, and there are optimal conditions, especially of things like osmotic pressure, under which the whole organism operates. All organisms have certain strict requirements. They have requirements for basic chemicals, out of which they're going to build their biomolecules. And those requirements are for carbon, various other things. Carbon is the most important because it's the backbone of all biological molecules. Then they also have requirements for nitrogen, sulfur, phosphorus, various other things. They also have a requirement for trace elements for very small amounts of certain things like metal ions, that sort of thing. Depending on the organism that we're talking about, they may have a requirement for oxygen. This is not universal by any means, but certain organisms definitely do require oxygen, and those are particularly, of course, those which use aerobic respiration, as we do. Lastly, in order for enzymes especially to work properly, the organism may require organic growth factors. Look back at the enzyme section and remember that the enzyme may have to bind a coenzyme of some sort in order for it to operate properly. That would be a typical organic growth factor. Okay, so let's have a look first of all at temperature, because temperature is one of the most important determinants for organisms. Now this is now discussing whole organisms, not just their individual enzymes, but the sum total of the metabolisms for different organisms has different optimal operating temperature. For the most part, what we deal with in clinical settings and in the lab and everything else are organisms which are called mesophiles. Here they are here, and you'll see that they have an optimum which is around about 37 degrees Celsius. Mesophiles, that's 37 degrees Celsius, of course, is very important, is in the pathogens and also in non-pathogens, which constitute our microbiome, for example. They are all very strictly dependent on a temperature around about 37 degrees Celsius, which is close to the body temperature of mammals. But there are a vast number of environmental bacteria which are also mesophiles. Their optimum temperature may differ a little bit from this, but in general, the mesophiles conform to having an optimum temperature around about 37. And then there is another critical temperature. It's called a cardinal temperature, which is the lowest temperature at which any metabolic activity will occur at all. And that in the mesophiles is around about 10 degrees Celsius. Now, when you take an organism down to that temperature, 10 degrees Celsius, is seldom fatal. The organism will survive, and if you raise the temperature again, then it will resume activity up to its optimum. So that is the first thing. The second thing is look, though, at this temperature, this cardinal point here, which is 48 or 49 degrees Celsius for the mesophiles. This is a little different. Just give me 1 second. I just want to see if we can get this thing to work. Okay, now you can go away. Go away. Come on, go away. Okay, so this point here is close to 50 degrees Celsius. Something different happens, but to understand why, cast your mind back to what we discussed about enzymes. That if we heated up enzymes, most of the enzymes in the body are proteinaceous. And at higher temperatures than the optimum, they very rapidly become inactive. They, in fact, degrade, and they cease to act, and they cannot be restored. That's individual enzymes. They become denatured at higher temperatures than the optimum, quite rapidly. And you cannot restore activity to an individual enzyme. The same thing applies to a whole organism. Take it above its optimum, and very rapidly, all of its enzymes and proteins become denatured. It dies. So although lower temperatures are seldom fatal, metabolism stops at the low temperature, but at high temperatures, the organism is killed and the activity cannot be restored. That's the typical shape for temperature tolerance of most living things. They have an optimum, they have a lethal higher temperature, and for bacteria, they have a temperature down here where activity stops, but is seldom fatal. There are other groups of, remember what I've told you often, that the bacteria are extremely versatile. And we see a whole range of different temperature tolerances in different in the different bacteria. Most, as I say, are mesophiles, but there are some which are able to tolerate much higher temperatures. Their proteins, the enzymes, are specially adapted to higher temperatures. And here you can see the optimum here. It's around about 60 degrees. Well, 60 degrees is really hot. You know, when those of you who had me in lab, I always tell you to heat your slides to fix them. And you put it on you tap underneath on your hand. You can just not hold it there, then the temperature is correct. That's about 65 degrees Celsius. 65 degrees Celsius, we fry most of our mesophiles. But here the thermophiles are able to exist in hot water. Water up to 60, 62, 3 degrees Celsius. That is really hot. Most living things are cooked at that temperature. Their enzymes simply can't survive. They do have a minimum. Look at the growth will cease around here, about 40 degrees. And they do have a maximum. They are denatured around about 72, 73. So they do have lethal limits, but their optimum is very, very high. And these are organisms, for example, that you might find in naturally hot water, for instance, thermal springs, those sorts of things. But surprisingly enough, a clever master's student at NYU, I think it was, went and looked in hot water cylinders all over the city of New York and discovered that many of the hot water cylinders actually have these thermophiles growing in them. So they don't only occur naturally, they occur under artificial conditions as well. Then there's a group which are extremely interesting. And these are called the hyperthermophiles. The hyperthermophiles, look at their optimum. It's anywhere from 85 all the way across, around about 95 up to 100, or 100 degrees Celsius is boiling. That is the boiling point of water. And yet here we have a group of bacteria which can survive that. And in fact, their minimum is down around 67 degrees Celsius. They do have a lethal higher temperature, that is true. But how is it possible that they are surviving in boiling water? How is it possible, in fact, that boiling water exists long enough for them to establish populations? Well, these are organisms which live in special circumstances, for example, in deep ocean waters, at volcanic vents in the deep ocean, where the pressure is so great that it actually prevents water from boiling. It can reach a temperature of 100, 110, but it does not actively boil. And very, very large populations of bacteria exist under those conditions. An extremely interesting group. Lastly, we'll have a look at those which have lower temperature optimum. This group here are called psychrotrophs. That is, they like cooler water. And these are organisms, for example, which are quite common in ocean water and in water which is naturally cold, especially, and in soils which are naturally cold as well. And the psychrotrophs have a temperature optimum, which is down around 21, 22 degrees Celsius. Well, that is not terribly cold. It is cool. It is certainly cooler than the 37 degrees that the mesophiles have. And we find that there are abundant populations of bacteria in these circumstances, in ocean waters, those are things who have this pattern, an optimum around about 21 degrees or so. Then there are a group called psychrophiles. Psychrotrophs are those which move towards cooler water. Psychrophiles are those which positively require a cold temperature. And in fact, their lethal upper temperature is really quite low. It's around about 17 or 18 degrees Celsius. their optimum is around about 10 to 12. And they can continue activity even well below that. And these are bacteria, for example, which you might find associated with ice flows, Arctic water, deep bottom ocean water, which is very cold. And these are the psychrophiles. You see that there's considerable overlap, especially in this group here. There's considerable overlap of habitat requirement. Okay, so this becomes important to us, especially in dealing with storage of food, for example, or introduction of pathogen into foodstuffs, because many of the organisms which might cause problems are in fact mesophiles. So for example, if food or water becomes contaminated with feces, most of the pathogenic organisms which might be transferred are mesophiles with a temperature optimum around about 37. Well, if we can lower that lower the temperature of the foodstuffs immediately, growth of those mesophiles will almost certainly stop at a low temperature. That is the reason we refrigerate food. That's why food is moved into refrigeration as soon as possible once it's manufactured. It limits the growth of pathogen, even if pathogens happen to occur there. So let's have a look here. Probably, this is the Fahrenheit thermometer. This is a centigrade thermometer. So have a look at our mesophiles, and you'll realize that their growth is encouraged at around about 37 degrees Celsius. But in some of them, it may continue even higher than that. And if you have organisms which are slightly thermophilic, it may continue even higher than that. Around about 50 degrees Celsius is about the upper limit for most mesophiles and most pathogens that we would encounter. And in fact, growth there would already be very slow. Any higher than that, they become impeded. But between 50 degrees Celsius and about 15 degrees Celsius, many bacteria will happily continue growing. And most of them, their optimum is around about here. So that's where they would be going bonkers if you were storing food at that temperature. This is a classic situation, of course, for things that have become contaminated, for example, with Salmonella, or even with Staphylococcus aureus. Both of these cause very rapid food poisonings, and they grow abundantly at this temperature. If somebody has an infected finger and makes potato salad, takes it to a picnic and sits it on a table for a couple of hours before people eat it at this high temperature, that could be a recipe for disaster, literally a recipe for disaster. Now, below that, as we start to cool food down and refrigerate it, most bacteria, of course, will survive refrigerated temperatures. There are a few bacteria of great importance, for example, one called Listeria, or Listeria, depending how you want to pronounce it, which actually actively grow at low temperatures, but most bacterial growth will stop here. So there are very few pathogens which will survive. There are bacteria which will spoil food, which will make it taste bad and look bad. That, as you probably know from storing food in the refrigerator, there are organisms which happily grow, if slowly, they will still continue growing at low temperature. Below zero, there's very little growth at all of any organism. There are very few organisms that do that. Okay, so similarly, there are optimum pHs at which organisms grow. Again, largely dependent on the pH requirements of their enzymes, but the vast majority of bacteria are in this group here. They're called the neutrophiles, and they grow mostly between 6.5 and 7.5. pH. So very much around neutrality. Molds and yeasts tend to grow a little bit lower, pH 5 and 6, but both these groups are eliminated usually by acid conditions. There are very few bacteria or yeasts of interest that will survive at lower pHs than this. Now, I mention this because there are situations in the body where we deliberately create acid conditions. For example, the vagina supports a population of bacteria which are completely normal in the vagina, and one of the groups that is supported in that microbiota are lactic acid bacteria, which produce lactic acid and lower the pH below 5. And this limits the growth of many other bacteria, which might be pathogenic, but it also limits the growth of yeasts. And this is one of the reasons why variability in the pH of the vagina may result in the blooming of yeasts, of very, very irritating, annoying yeast infections, which are normally kept at bay by lower pHs. So most bacteria, the vast majority, are neutrophils there. But there are some groups which grow at much lower pHs, and these are called the acidophiles. Look at these here. Their pH optimum around about 3. Okay, so where do you think these might grow? There are two particular situations where we see this kind of pattern. The first is environmental. When we create waters, for example, which are highly acidic, one of the situations where that occurs is in mine drainage. Water draining out of mines very frequently has a low pH, and that encourages the growth of acidophiles. The second is a natural situation. And take a couple of beats of your heart to think about it. Think of one natural situation where acidophiles might grow, for example, in your own body. Apart from the low pH vaginal microbiota, the other situation, of course, is in the stomach. The stomach has a very low pH, and it actually still allows the growth of certain bacteria. Your stomach actually has bacteria which are able to survive at this low pH. There's actually a pathogen which is able to survive at this low pH, and it's called Helicobacter, and it is the cause of stomach ulcers. And it survives because it has special adaptations, which we'll discuss later on. It has special adaptations, and it can survive higher pHs, certainly, but its optimum is down around 3. Similarly, there are groups which are able to survive at very high pH in highly alkaline waters, and these are the alkalophiles. One of the strange things in prokaryote biology is that these groups, which can survive very high pH and very, very low pH under natural conditions, under normal environmental conditions, I should say, are very often members of that group of prokaryotes, the archaea, which are the other prokaryote group apart from the bacteria. The archaea very often show extraordinarily high ability to survive harsh conditions, not only high and low pH, but also very high temperature. And as we'll see in a minute, they can also survive very, very high osmotic pressure. They can survive in solutions which are absolutely saturated with salts, which no other bacteria can do. Okay, so let's have a look now at the importance of osmotic pressure, because it's one in biological systems, it's one of the most important determinants, and it's something that all cells have to deal with. All cells exist in some sort of an aqueous medium. This is a characteristic of life. Our own cells are bathed with body fluids. Many, of course, you look in the environment, they are in suspension, in solutions, etc., etc. So the cell itself, remember, the cytoplasm of a cell is a complex solution, a complex mixture of molecules, usually in very, very much higher concentration than occurs on the outside. The correct way to think about this is to think about the concentration of water. So if I have a cell and its cytoplasm is full of molecules of all different sorts, there is literally no room there for water molecules. So you need to think this high concentration of salts within the cytoplasm means that there is a low concentration of water. Most organisms now have to deal with the fact that by diffusion, the outside of the cell usually has a much higher concentration of water. So it's going to move from high concentration to low concentration. Most cells have to deal in one way or another. Organisms have to deal with the fact that water is going to move into the cell continuously. There are different ways of dealing with this, but let's have a look at a simple example of what happens under different osmotic conditions. So we're going to discuss this in terms of the solution on the outside. Okay, and you'll see the importance of this in a second. Get it clear in your mind. We are talking about the concentration of water in the solution around our cell. If the concentration of water is the same outside as it is inside the cell, then we say that that is an isotonic solution. It is isotonic with the interior of the cell. This has an important effect. Water will still pass across the membrane. It will still pass by one means or another. Water can still pass into the cytoplasm, but think about this. Just think in terms of one water molecule. We've got an isotonic situation. The solution is isotonic to the inside. The water concentration here is exactly the same as it is on the inside. Let's take one water molecule, pass it into the cell. This is no longer an isotonic situation, because now the interior of the cell has one more water molecule than there is outside. What will happen? By diffusion, one water molecule will move out. That is the definition of an isotonic solution. The concentration of salts, etc., and hence the concentration of water is the same outside as it is inside. And the passage of water molecules into the cell is balanced by the passage of water molecules out of the cell, so that isotonic conditions are maintained. This is largely what we do. We bathe our cells in a solution which is kept isotonic by our kidneys. So we bathe our cells in isotonic solution so that we don't have to expend any energy dealing with osmosis, causing problems for ourselves. The passage of water molecules, the diffusion of water molecules is specifically referred to as osmosis. So that's a relatively easy situation to understand. Let's move to a situation where the outside of the outside solution has far fewer dissolved salts, etc., than the interior. Well, what is our situation now? What is the concentration of water molecules outside and the concentration of water molecules inside? This is a situation which most environmental cells face. They're almost always in a solution which has far fewer salts dissolved than are dissolved into the cytoplasm itself. In other words, the cytoplasm has far less water than there is outside. And the diffusion drives water into the cell. That diffusion of water is osmosis, and it comes in at a certain pressure. That is the osmotic pressure. The osmotic pressure is the pressure which drives the water molecules into the interior of the cell in an attempt by diffusion to balance the concentration outside and inside. Now, the cell is in great danger because the passage of water into the cell means that the cell is going to start swelling. And if that carries on much longer, this fragile cell membrane is going to burst. And this is a very dangerous situation for a cell. And it's a situation which many cells will face, usually especially in environmental situations. The cells are in solution, which is hypotonic to the interior of the cell, and water passes rapidly into the cell, and the cell has to be adapted to deal with that. They deal with it in various ways. Experimentally, if we were to take something like a red blood cell, as we used here, put it into fresh water, very soon you can actually observe the red blood cells swelling and bursting due to the passage of water in. A different situation occurs if we take a red blood cell or any cell and we put it into a solution where the outside is very much more concentrated than the inside. Now, there's a higher water concentration inside the cell than outside, and water will pass out, and the cell will begin to shrivel. And this is also a situation which many cells face from time to time, and it's something that they have to be adapted to. We'll mention one in particular. Okay, so remember, we always describe tonicity in terms of a relationship. Outside, here we say the outside solution is isotonic to the inside is isotonic to the outside. Here we said the outside is hypotonic to the inside is hypertonic to the outside, so on and so on. We always describe in terms of the relationship of the two. This is hypotonic to the inside of the cell because it has a much higher water concentration. So let's have a look now in bacteria and see what the impact of this is, because bacteria can move from one situation to another, especially environmental bacteria, may be subjected to different osmotic pressures. Usually, bacteria are exposed to an environment where the outside is hypotonic. That is the normal situation. The outside has far less salts and everything dissolved in it than the interior of the cell. Bacteria therefore have a danger that they would continue to swell until they burst. But they are protected from that by the presence of the cell wall. There it is there. We've heard ad nauseum about the structure of the cell wall, the peptidoglycan, etc., etc., etc. This is what it's there for. This is its principal role. It acts as like a wickerwork skeleton around the cell, and the cell can swell due to osmotic pressure, and it will swell up against the cell wall. And you'll realize that what is happening is we are actually building up pressure inside the cell. Now, when the cell swells against the cell wall, the pressure builds up until the interior pressure, which is also referred to as turgor, is equal to the osmotic pressure. And now, as many molecules of water pass in, have to pass out, because the two pressures must remain equal to one another. So that much water passes in, passes out under this situation once the cell has swelled to maximum pressure against the cell wall. That is the principal function of the cell wall. It does also give shape to the cell, etc., but it is mainly an osmotic protection. And bacteria can also exist in isotonic situations. That is true in body fluids, for example, in the gut, etc. They are largely in isotonic. But in a hypotonic solution, they swell up again, the cell swells to the cell wall, and then swelling stops. But cells may also be transferred to hypertonic situations. And under those situations, for example, if you take a normally adapted environmental bacteria and you drop it into a solution which has a high number of salts, here they are using sodium chloride at 10%, which is very high. What is going to happen is the water is going to flood out of the cell. It's going to flood out of the cytoplasm to try and dilute the outside by osmosis. And what we see then is the cell begins to shrink away from the cell wall. And this is called plasmolysis. It's a very dangerous situation. And very often if plasmolysis continues too much, the cell dies. This is very useful. It's very useful for human beings. Why? Why do we salt food? To preserve it. Think of salted fish, salted vegetables, sauerkrauts, these sort of things. We can actually create an environment where for most bacteria, they will plasmolyze and die. And they won't be spoiled and they won't grow pathogens. And that's how we use salt to preserve our food. Okay, so I will also mention that there are, again, remember, bacteria do absolutely everything, and archaea as well. There are a certain bacteria and many, many archaea which are obligate halophiles. That is, they can only grow in a situation where there is a very high salt content around them. And this is particularly true of the archaea. They grow very well in areas, for example, like natural salt pans or artificial salt pans. If you've ever flown over the San Francisco Bay Area, you look out the airplane window, you'll see there are still salt pans, commercial salt pans there, which are often very brilliantly colored red, purple, bright green. These are extreme obligate halophiles growing there in the salt water. There are others which can tolerate a high salt content for a while, but they only tolerate it. They survive there, but they don't grow vigorously. But they can be taken down to a lower concentration of salts and they will happily grow again. So just remember that word, halophile. That means either wanting or tolerating high salt contents. I won't go into detail about it, but I will tell you that they have very special cellular mechanisms whereby they maintain an interior environment which is favorable for metabolism. Okay, so that deals with kind of the environmental situation for cells. Now we need to talk a bit about what cells actually need to grow. Well, the first thing you know about, and that is carbon. They have chemical requirements for various atoms, various molecules. All cells do, but the primary requirement is for a source of carbon. And that is because all of our molecules, our biomolecules, are structured on a skeleton of carbon molecules joined to one another. And that carbon forms the structural backbone for all of our molecules. There's the group to which we belong, the chemoheterotrophs, also require an already created organic molecule as a source of carbon, but also as a sort of energy, as a source of energy. And that group to which we belong, and we are called chemoheterotrophs. We depend on molecules already created, biological molecules already created, as a source both of carbon and other molecules, and as a source of energy. But the autotrophs, the autotrophic organisms, whether they're photoautotrophs or chemoautotrophs, go back to what we talked about in the last class, are able to use carbon dioxide as a source of carbon. They don't use carbon dioxide as a source of energy. They use it purely as a source of carbon to build the initial molecules, usually sugars, which then fund all of the rest of their metabolic needs. In addition, the next most important atom that we need, element that we need, is nitrogen. And that's because the nitrogen is a component of so many different molecules in the body, such as the proteins, DNA, all of the things which are based on nucleotides like ATP, GTP, all of these things require the presence of nitrogen. It's a very, very important element in living systems. And most bacteria are actually able to decay already existing complex molecules, such as proteins, etc., as a source of nitrogen. But remember that ultimately, nitrogen is actually returned to the atmosphere, and usually in the form of a nitrous oxide of some sort, or a nitrogen oxide, sorry, of some sort, or else as free gaseous nitrogen. We've already heard one instance where this occurs, and that is where bacteria are able to use a nitrate-rich compound as a source of oxygen for respiration in the process of anaerobic respiration. Under those circumstances, eventually, all of the nitrogen built into organic molecules is liberated into the atmosphere. And there's something very bad about that. And that is nitrogen is very, very inert, especially when it's in the dinitrogen form N2, it's practically inert. It's very, very difficult to get it to react with anything. So it's very difficult to get it to re-enter biological cycles. And here we depend solidly upon a group of bacteria which are able to fix nitrogen. That is, they can take atmospheric nitrogen and they can put it into organic molecules, which can then be used both by the cell itself, but then by other living things as well. All right, and there are a couple of other elements we need. Sulfur is really important because sulfur is used in amino acids in various vitamins. And it's in amino acids, for example, the presence of sulfur enables proteins, which are polymers of amino acid. It allows, in proteins, it allows sulfur groups to bind together and to help structure the structure of the protein. And there are many ways in which sulfur is used in biological systems. And one of the reasons that bacteria, for example, decompose proteins is not only as a source of nitrogen, but also for the sulfur that they contain. And there are other bacteria, autotrophic bacteria, for example, which can use sulfate or hydrogen sulfide as a source of energy. Phosphorus is the next important element used in DNA, RNA, and various things that are phosphorylated. And it is very often, those phosphate groups, as we've heard before, are very often a way in which to make energy available. Transfer of phosphate groups may, along with it, transfer energy from one substance to another. Quite apart from that, the whole structure of the membrane, of the cell membrane, for example, it depends upon the presence of phospholipids, of phosphate groups bound to lipid groups. And that very often in biological systems, in fact, The phosphate groups are a major source of phosphorus. Phosphate groups extracted from food, etc. Well, bacteria also often require the presence of certain already created organic compounds. It depends on the bacteria. Many bacteria are able, if they're provided with a source of all of those elements, nitrogen, phosphorus, sulfur, etc., etc. If they're provided with an inorganic source of all those elements, they are able to create all of their necessary molecules. That is not always the case. Sometimes there are organisms which are unable to manufacture all of their required organic molecules, and if they do so, those that they require we refer to as organic growth factors, and they have to obtain them from the environment, from food or whatever. They can't create them themselves, and a wide variety of things. Vitamins, amino acids, various nucleotides, the purines and pyrimides are nucleotides. So it depends on the organism what their requirements are. But you always have, if you're growing bacteria in the laboratory, for example, you have to be aware of what the external requirements of that organism should be. Then the last thing to talk about are the trace elements. These are inorganic elements, often metal ions, for example, which are required in very low quantities. I'll give you one good example that should be fairly obvious, and that is iron. Iron is a trace element that we require because it is part of the heme molecule in hemoglobin and various other substances as well. It is just a single iron atom sits in the middle of a very complex proteinaceous molecule which forms hemoglobin. Just one iron molecule in the middle guarantees that is actually how hemoglobin works. It's through that one iron molecule. But there are various other metal ions which are also required, often as cofactors for enzymes to operate. And remember that an enzyme cofactor is an inorganic requirement for the enzyme. And these are iron I've told you about. Hemoglobin, by the way, is an enzyme. Others, copper, molybdenum, zinc, various other ions. Boron is one of them, in very low quantities, and usually easily obtained from the food sources, nutrient sources. Okay, so now comes one of the most important environmental considerations that we need to talk about, with bacteria especially. And that is the relationship of the organism to oxygen in the environment. Now, in the bacteria, we see a very wide range of relationships to oxygen, as you'll see in a minute. Experimentally, we can demonstrate these relationships quite easily. And that, what we're going to do is we're going to set up tubes of loosely solid agar, that is very low concentration agar. And we're going to include in this agar a substance which will actually bind oxygen. So in low concentration, we're going to put that in low concentrations. If we just take a tube like that, we boil it, drive off all the oxygen, and then leave it to the atmosphere, what we'll find is that it will build up a high oxygen concentration up here, a much lower here, and an absence of oxygen here at the bottom. So just excuse me 1 minute. Oh, my cat is yelling at me through the door, but I just try to ignore him. Okay, so when I teach from home, you've got to put up with all of these things. People at the door asking for money, my cats leaping and springing outside. let's have a look now. We've got our tube, high oxygen, middling oxygen, very little or no oxygen at the bottom. Now we're going to inoculate these tubes. We're going to inoculate them with various different kinds of bacteria, which have different responses to the presence of oxygen. And we're going to see how they grow and why that occurs. The first group we're going to put in are called are members of the obligate aerobes. Obligate aerobe, they positively require the presence of oxygen in order to grow. And there are many of these, especially occurring environmentally. When we inoculate this tube and we leave it to grow, we'll observe growth only at the very top there. And there is a reason for that. They positively require oxygen. They are non-fermentative. They cannot resort to fermentation as associated with glycolysis in order to survive. They just can't do that. They are entirely dependent on aerobic respiration. These bacteria have to contend with another feature of oxygen load, and that is oxygen itself is extremely dangerous. It's a very dangerous and highly reactive molecule, which it will actually interact with biological molecules and break them down. So these organisms have another thing to contend with, and that is that aerobic respiration creates a certain amount of strange oxygen species. That is, they first of all create superoxides, 3 oxygen atoms bound together, which is highly, highly reactive. And they create also single oxygen atoms, which are also highly, highly reactive. And the cell has to have ways of dealing with those. Otherwise, they damage molecules inside the cell. There are 2 very important enzymes, which these strict aerobes, obligate aerobes, produce. And these are, first of all, the enzyme catalase. Catalase breaks down one product of aerobic respiration, which is peroxide. That is hydrogen peroxide, H2O2. And that is also extremely reactive, extremely damaging, and it is broken down by catalase to oxygen, dioxygen molecule, and to water. And the second is superoxide dismutase. Superoxide dismutase breaks up those groups of 3 oxygen atoms and neutralizes. So they produce both those. Catalase and superoxide dismutase destroy these toxic forms of oxygen. Our next group that we're going to try inoculating into another tube are the facultative anaerobes. And the facultative anaerobes can grow aerobically perfectly happily, and they do so extremely efficiently. They perform aerobic respiration and are able to grow at high metabolic rates. But if we deprive them of oxygen, they can continue to survive and to grow to a certain extent by resorting to fermentation associated with glycolysis. The type of fermentation that they perform depends on the organism, but they basically use some form of fermentation associated with glycolysis, as we saw before. Go back and revise what we talked about. So when we inoculate them, what we find is they grow abundantly up at the top of the tube, but not so well through the rest of the tube, but they can survive. They will grow to a certain extent. They do produce both catalase and superoxide dismutase, depending on the bacteria, I should add. But in general, they will produce both of these enzymes. So in many ways, their behavior is similar to that of the obligate aerobes, except that they can survive by fermentation and in the absence of oxygen. The next group are extremely important to us clinically, and these are the obligate anaerobes. Obligate anaerobes lack any enzymes to neutralize harmful forms of oxygen. So they have no catalase, no superoxide dismutase. And they are absolutely unable to survive in the presence of even of extremely low concentrations of oxygen. In our tube, they would be isolated to the very bottom here. Growth would be isolated there. Why do I say that that is so important clinically? Well, most of the Clostridia, for example, Clostridium is an organism which is very important clinically. It causes various diseases. For example, it causes gangrene in wounds which have gone anaerobic. It causes a condition called Clostridium difficile, which is a gut infection. Again, and it has to operate under totally anaerobic conditions. And unsurprisingly, one of the ways of treating one of these, an infection with one of these organisms, is to put people into a chamber with a very high pressure of oxygen, called a hyperbaric chamber, in an effort to saturate their tissues with oxygen and kill off the obligate anaerobes. Just on this note, clinically, I just want to say something to you. I often get asked, like, how is it that Clostridium entering a wound, for example, in a human being, can grow when our tissues are full of oxygen? We breathe oxygen the whole time, we move oxygen around, we use it as our terminal electron acceptor. The answer is that there is very little free oxygen in our tissues. Most of the oxygen is bound to hemoglobin. It's abundant oxygen in our lungs, that's for sure. But oxygen is moved around our bodies in a bound form and then is utilized by taking oxygen from that bound form directly into the cytoplasm of tissues. Our tissue surroundings do not have high oxygen levels. They may have no oxygen levels. That is how, if we spike ourselves with something which is contaminated with an obligate anaerobe, the wound may grow anaerobic bacteria. Once they are growing, by the way, then they may create their own anaerobic environment, and they may reinforce that anaerobic environment. There's another group called the aerotolerant anaerobes, and I would just want to, this is really important. There is a mistake in this diagram. That word there should be taken out. They will, and I'll explain why. The aerotolerant anaerobes only grow under anaerobic conditions. So they will grow abundantly down here, but they will survive the presence of oxygen. These cannot survive the presence of oxygen. These can, but they don't grow in the presence of oxygen. They will only grow in the absolute absence of oxygen. And the reason for that is they only have one of the enzymes, superoxide dismutase. They are unable to deal with abundant oxygen, but they just shut down. They stop metabolism as much as possible until they move into totally anaerobic condition. The last group are called the microaerophiles. And the microaerophiles are those, they need a very low concentration of oxygen. They grow best where there's a very small concentration of oxygen. And their growth is actually anaerobic. Is actually aerobic, but it is extremely slow and does not proceed well in high oxygen levels or in the total absence. And actually, the reason for that is they have very low resistance to toxic forms of oxygen, but they are able to tolerate a certain amount. Okay, I'm going to stop there for today, and then we will continue with the short bit that remains after your exam on Monday. Have a good weekend.